Okay, so last year I spoke about convolutional neural networks, and that was pretty basic. I think now we're all using them or know about convolutions, and you probably heard it in previous talks. And I think indeed a topic that I thought would be very interesting is, given that we have a great predictor now that performs very well, how do you interpret and learn some biology from it? So here, just in this figure, I show you some CNN from the DeepBind paper, convolutional neural network, another network combining convolution and recurrent neural networks. And here is just a hint of what I'm going to talk about, which is the interpretability and how can we learn something about biology. By the way, if you want, you are welcome to cite our review paper that was published in Annual Reviews of Biomedical Data Science. So some promotion for my work. Okay, so let's start with a sort of discussion and asking the crowd, why do we care about interpretability? I mean, we build a predictor based on convolution, recurrence, transformer, hidden Markov models, get Pearson correlation of 4.9, AUCs of 0.99. You can't beat us. We've got the best predictor. Why do you care about interpretability? And that's a question to the crowd, if anyone wants to chip in and contribute. Yes, I don't know. Yeah, please go ahead. Someone raised the hand.
Yeah, perhaps just simple. It's always nice to know what is going on. And we already yesterday someone said the deep neural network is generally a black box, and the same for many others machine learning methods. And even if we know our prediction is working in training and test sets, perhaps with a new test set it could be small differences, and then we would like to know why. And so there would be the interpretability helping us.
Right. And I would even say it's not just nice, it's critical to know what's going on. Okay, so thanks for the answer. So indeed, having an accurate predictor, highly accurate predictor, is just one requirement, right? It's a great tool, a black box, as we just heard, that can give us the option the ability to generalize what the data, what the principles that we learned to any new data point, any new DNA sequence, RNA sequence, or protein. But indeed, interpretability in bioinformatics is already a requirement. Any study that you do, research or paper you submit, must have a point where you ask the computational model what are the principles, the biological or molecular principles it can. So that's something that you need to learn, and that's why I think having this set of computational techniques is critical. And I think in bioinformatics, it's not only that we are here to develop tools, provide a tool for the researchers, for the biologists. We also want to learn biology on the way, to do some science, and to get insights behind the biological phenomena. So indeed, interpretability is something that has been solved and tackled for years, for decades, since we worked with expectation maximization and applying to simple matrix models or hidden Markov models or whatever. But what we see in the field in the last 5 or 6 years, right, is the swapping of deep neural networks over any problem that is solved in genomics with high-throughput data. And that requires new interpretability techniques. It's not just we can use the same old techniques that we have been using in previous decades. We need to learn new techniques. And that's what I aim to do in this talk, to give you some of the lead techniques in the field to interpret deep neural networks. And please, if anyone has a comment or objection, you're welcome to share them. Okay, so I'll start, in order to learn these techniques, I'll start with what we've been doing in previous decades and talk about position weight matrices and sequence logos. And these will be important as we go into visualizing deep neural networks, and specifically with convolutional kernels. And then I'll also explain the technique of mutation maps. And then I think the great tool of integrated gradients, it's I find the most useful one when I apply. in my studies. And if I will have time, I will also talk about TF Modisco, which is a tool to find common patterns, sort of doing motif finding on attribution maps. And if I have time, I will explain it at the end of the talk. So let's start with the basic, what we have been doing previously. So maybe someone wants to answer, what is the most popular model that is used or has been used to model protein DNA and RNA binding? Anyone wants to guess? Okay, so I will give you the answer. That's the position weight matrix. And a position weight matrix is here, for example, a matrix of probabilities where each position in the matrix is a distribution over A, C, G, and T, and gives us the probability of observing that nucleotide in each of the positions. Okay, so now we can score any subsequence of length 9 and just multiply the probabilities and get the probability of observing that subsequence. And these probabilities can be converted to weights. One of the issues in computation is that multiplying probabilities will get us to very small numbers. It might lead to numerical issues in the representation and the computation. So we can apply a log transformation and convert these numbers to a log scale. And now in order to get a score for a specific subsequence, we can sum up the weights. And here is an example where we have a sequence. We want to score a subsequence. We can score all of them by a sliding window approach. And we can sum up, add the scores of each nucleotide in each of the positions. For example, C here would get a 5, T here would get a 5, and so on. And the reason I'm covering the position weight matrices is because they have a very interpretable visualization. Actually, one of the reasons why the model is so popular, in addition to its simplicity and effectiveness, is the fact that you can visualize it in a very interpretable way.
The reason that you divide it by 4 is because you're assuming a uniform distribution between the nucleotides?
Okay, so excellent question about this division by a quarter. The log transformation can also take into account some background probability that is expected. So for example, here it's a uniform distribution of A, C, and GT, but you can also think of a region that are GC rich, for example, promoters in mammalian genomes, and you will divide it by different probabilities. Maybe the GCs will get 0.3 and A and T will get 0.2. So you can take the background probability into account when you do the log transformation.
Excellent, thank you.
So I will explain in detail and give the mathematical definition of a sequence logo in the next slide. But before I move on, let's ask the audience, what is the equivalence in deep neural networks to a position weight matrix model? I can give a hint. It's one of the building blocks in a convolutional layer. So the answer would be that in a convolutional layer,
Perhaps.
Excuse me, yeah?
The kernel?
Exactly. A kernel is a matrix. If we apply a one-dimensional convolutional layer on an input sequence, a kernel is equivalent to a position weight matrix. We learn weights, and each time the kernel moves along the sequence, it sums up the weights and assigns a score in that offset of the sequence. So, and of course, the kernel weights are not constrained in any way when we do the training, as opposed to a frequency matrix where we have these constraints. But if we go to the log scale or unconstrained weights, then it's exactly equivalent to a kernel. And that will be a connection we will use later on. So let's learn about the definition of a sequence logo that we see in many of the studies, and it's a great interpretability tool. First of all, let's say we are given a probability frequency matrix. Okay, so our input is this base probability matrix. The length is some length that we decide to, it's a parameter of the model, and we get a distribution over the nucleotides. Then my question to you, what is the definition of the position height? For those who know, are welcome to share. What's the equation formula to compute the position height? And maybe I can give you a hint. It's also called the information content of the position. Okay, so another question, what would be the entropy?
I mean, Shannon entropy, I think.
Right, so what is the entropy? That's the sum over pi log pi,
No,
right?
Yes, Shannon entropy is some information about uncertainty, I mean how all the information is exist in the sequence. So the formula is yeah.
Yeah, do you know the formula?
You mean the HI is sum of FBI in log FBI usually.
Okay, that's good. So the formula for the position height will take the entropy, and if I'm correct, it's minus of the sum of pi log pi, where pi, the sum goes over ACGT, and adds 2 to it. So it's 2 minus the entropy. And that's the formula to get the height of each position, also known as the information content. And you can use this formula to remove uninformative positions in a position frequency matrix. So this really captures how much uncertainty or certainty we have in a position, right? Here we see a position that is most likely to be an A, while this smaller, lower positions are more uncertain, are more closer to the uniform distribution.
Why is the 2 chosen here? Why is it 2 minus the entropy and not, say, 1?
And not 1?
Yeah, for example, it's just.
Okay, so let's say I tell you that ACGT distributes uniformly, and I send you a nucleotide. How many bits did I send you? As bits of information.
4, because we have 4 nucleotides, or 4 times the information.
Okay, so to answer that question, if imagine that the probability is 0.25 and I send you a nucleotide, is to take the alphabet size and put it in log of base 2. That gives you the number of bits of information, right? It's as if we can convert A to 0, 0, C to 0, 1, G to 1, 0, and T to 1, 1. So when I send you a nucleotide, you get 2 bits of information.
Okay, I see. Thank you.
So that's why the scale here is here between 0 to 2. Okay, and now the distribution inside the height depends on the frequency. So for example, here, if A is maybe 70% chance and Z is 30%, we get this distribution inside once we decide the letter height. Okay, so now given this formula, I have a few questions for you. Which position would achieve 0 height?
Equally distributed?
Right. If we decide to put 0.25, the uniform distribution in a position, you see here that we take a quarter times log of a quarter in a base 2. What's log of a quarter in a base 2? That's -2. We sum it 4 times multiplied by a quarter, you get 2 minus 2, and that's 0. So as lower as the height, it is as close it is to a uniform distribution, meaning this position is probably uninformative, a position that the protein, if it's binding DNA, doesn't care about much, can tolerate any nucleotide. Next question, which position achieve a height of 2 bits?
When you know with certainty which nucleotide you receive?
Right? Exactly. So thanks for the answers. So if we have one nucleotide with probability 1 and all other 3 have probability 0, what we'll get here is 2 plus, then 0 times log 0 would be 0, and 1 times log 1 is also 0, so we get the height of 2. So here, for example, in this position where you see the A almost reaches 2, that means that it got very high probability, almost probably more than 90%. And then the last question is to give an example of a position which will achieve a height of 1. Right. If we have, for example, a probability of G and C 50%, and then A and T and 0, we get exactly the height of 1. We can put here the half times log half. Log half is -1, so we get 2 minus 1. That's in total 1. So if we have 2 positions with 50% and the other to 0, that gives us a height of 1. Any question on the sequence logo representation? Okay, so thanks for the participation. Another option to visualize the position weight matrices is by affinity logos. And these are simpler to understand. Here we, maybe you can guess how do we decide on this visualization? How do we convert a position weight matrix to this visualization? So the way, yeah, please.
With the formula that we had earlier with the normalization, then we expect the log of each probability and their sums, because I see that they sum up to 0 here. And
okay. I mean, given that I already give you the position weight matrix, so imagine that it's already past the log transformation. Now my question is, how do I convert it to an affinity log?
Just put the scores on the y-axis, no?
Yeah, the letter height is just proportional to the nucleotide weight. So it's much simpler, right? We take the weight from the matrix and then the height. And as you see, we can have both positive weights and letters above the 0 line and letters below the 0 line, negative. And here, for example, it's if the weights were representing some energy values in delta G, we can easily plot them using an affinity logo, but the weights can be anything. It could be kernel weights or from the log transformed position frequency matrix, and so on. So the sequence logo and affinity logo are two popular ways to visualize these matrices. And now I would ask, so I presented the sequence logos, what would be the most straightforward application to deep neural networks, or specifically to convolutional neural networks?
Using them as kernels?
Yeah, right. We can just take a kernel and visualize it, right? A kernel is a position weight matrix. We can take an affinity logo that I just presented and visualize it. But apparently, it works really bad. I don't know if you have seen in papers or have done it yourself, but it doesn't work very well. And now I can, maybe you can guess why does the application of these visualizations on kernels don't give us interpretable signals?
Because we have many kernels and one of them just doesn't tell us anything.
Right, exactly. So one issue is the fact that we have multiple kernels, and I think many. I think if you read papers these days, you will have hundreds of kernels in a network. And then what happens when we have so many, it's not that a single kernel captures an element, a biological element, but they might be sub-signals that together form some element on the molecular or biological level.
Shouldn't you also, sorry.
Yeah, please.
Shouldn't you also consider the reading frame of the sequence, or it's just a kernel based?
So are you referring to a coding sequence where we have different offsets?
Exactly.
Okay, so I'm not specifically talking about coding sequences and their offsets, but it's an issue that I will not cover in the talk today. But yeah, it's something that you should consider when you look into coding sequences and want to capture the right offset. But I don't have experience with that, so I can't comment more on what are the common techniques to solve it. And another issue is, given that we have a kernel and its weights, if you want to visualize it as a sequence logo, we need to convert it to a distribution. And it's really not clear how to convert a vector of weights over ACGNT to a distribution over ACGT. And the way we convert could turn into different visualizations. And here is one example that they give you. You can later tell me if it's a bad example or good example, or tell me now, but after I'll show it. But I've downloaded it from the DeepMind study. They have a database. I put in the GATA transcription factor, and here you see the visualization they provide. So you see GATA appears quite nicely here in the sequence logo, but it appears again and again and again. So there is some redundancy in here as well. And there are also many uninformative sequence logo, right? They are just a bunch of uninformative positions. They mean anything. So this is what I wanted to give an example why visualizing kernels doesn't work that well. Okay, so we have redundancy, since these outputs of the kernels, once applied on a sequence, they have some cooperation interaction, which we don't capture when we just visualize the kernels. So here is the complete procedure that was used in the DeepMind study. I want to cover it as the first algorithm in the field that was used to visualize kernels. They generate a position weight matrix from a filter response. They take all the test set sequences, both positive and negative, if it's a classification problem, through the convolutional and rectification stage. Just as a reminder, in the DeepMind network, they first apply a one-dimensional convolution layer, and then on the outputs of each kernel, a rectification stage is applied, where the rectification has a specific parameter, which is the threshold. So some of the kernel outputs pass the threshold, and some don't, and are set to 0. All the sequences, subsequences that pass the threshold are aligned, and these are used to produce a position frequency matrix just by counting the nucleotides in each position. And then we can plot a sequence logo as I told you a couple of slides ago. So here is the algorithm for each kernel. Only take sequences that for some kernel there is a positive output. If all of the outputs don't pass the rectification and set to 0, the sequence is removed. For each such sequence, they took the subsequence where the kernel produced the maximum output. And then this sequence was aligned with other subsequences that were applied on the other sequences. And then it's easy to count nucleotides and produce frequencies. And we can also trim the low information position. As I told you, we have the information content criteria that we can use. And here are the examples shown in the paper. So for example, this is a known sequence logos. And these are the ones they produced from a CELEX experiment, where CELEX is an in vitro experiment, and from ChIP-seq, which is in vivo. So they show that in the chipsec, they also found another motif, which is expected because in the in vivo environment, in a cellular environment, the binding signal is much more complex. Here, for example, they show this is the known binding site, and they recapitulated the binding site and also produced some artifacts of the experiment. And for the chip, they produce other two binding sites. And the list goes on, and there are many, many nice examples of what was produced by this visualization technique compared to what was known in the literature before. But as I told you, you can now go to the DeepBind database, pick your protein of choice. For example, I took the one that was just shown in the example before. And here you see that while in the paper they show the top three ones, the nice logos they created, there are many more uninformative sequence logos. So just to conclude on visualization of kernels, it still produces redundant and many uninformative motifs. It's unclear how to choose one kernel. And this is especially becomes a problem when networks these days apply hundreds of kernels in the first convolution layer, as opposed to the DeepBind paper, which was the first one in the field that used only 16 kernels. So that will lead me to the next visualization, which is the mutation map. Any questions up till now? Okay. So
sorry, maybe
yeah.
if you could introduce a little bit of terminology, what you are actually problem that you're approaching with the, like, what is, like, how to formulate the binding problem, because I'm not really from this field.
Okay. So yeah, maybe I jumped a bit and I assumed some prior knowledge, but the DeepBind study, if I go again, here I hinted at, solved both regression and classification problems. And the classification problem that was solved was a set of positive sequences that were known to bind by a specific protein. And then they added a set of negative sequences, DNA sequences, let's say of length 100 nucleotides, that were not bound by the protein. So that generated a binary classification problem of a network that needed to distinguish between positive examples bound by a protein and negative examples unbound by a protein. And that's why when I next refer to a protein, a specific protein, the kernels are supposed to represent the binding site, the region and the preferences where the protein would like to bind in a long sequence.
Thanks.
Okay, so I hope it clarified some of the prior knowledge, which I assumed and correctly I didn't give. Okay, so now let's move to the mutation map visualization. I think also already the term mutation map gives you a hint on what happens with this visualization. So given an input sequence of length n and a model, what we want now to do is to ask what would be the effect of each possible mutation on the sequence, given that we have the model. So we visualize the sensitivity to mutations by generating a 4 on n matrix. In our case, the alphabet size is 4, but of course we can apply it on amino acids where the alphabet size is 20. And we can also show the original sequence and assign a height to each letter by the sensitivity or how much the score of the prediction decreased. And I think a figure is worth 1,000 words. So here is an example. So the matrix, the heat map that you see below is the mutation map. It's a matrix on 4 on n, where for each nucleotide in the original sequence, for example, here we had T, we mutate to A, C, and G, and for each of the mutations, we use the model to predict the score that would be assigned. And now we can visualize here, we get a number, which is how much the score decreased or increased. So the blue color here is for how much it decreased, while the red is for increase. And we can plot these values in a heat map. And then also assign a height to each letter based on, for example, the maximum in this vector. And the reason, for example, maybe I can ask you, why, if you look at this example, the heat map, why do we have a single white box in each position, in each column?
Because that's the original value of that sequence.
Yes, okay, exactly. So for example, here you see a T. If we mutate the T to a T, of course, that's the original sequence, the difference would be 0. So that's why in each position, you will see exactly one element that is of value 0. So here, for example, you see if we mutate this C to A, G, or T, we get a huge decrease in the binding score. While other position outside of the binding site, we almost see no decrease, and they are quite white. If we look at a mutated sequence where the C is a G, and the and the G increase is changed to a C, this gives us back the original binding site. And here is another example of a binding site, and it's the effect of mutation. Here, for example, there is a position that mutations to A or C would even increase, create a binding site that has higher affinity by the protein, and a mutation of this position from a G to a T has some effect, but still keeps the binding site. For example, here in the mutation from a C to a G, it really destroyed the binding site. Here we still have some of it. So I will give you the formula that was used in the DeepBind, but you can think of other formulas and ways to create this mutation. So we start with computing the original score, computing the score of the original sequence, denoted by P of S. Then we generate all possible mutations. So we replace the nucleotide at position I with any possible nucleotide J out of A, C, G, and T, and we generate S at as the mutated sequence. Then we compute the P of S at, and we can define the sensitivity as the difference between them, right? P of S at minus P of S. How much the score decreased or could also increase. Now, in the DeepBind study, they decided to add another term here, and I will give you the motivation. And the second term is to design to ignore variations in the no binding. For example, if P of S and P of S at are both negative, then maybe we don't care about the differences in that regime. So the max is 0. Or if P of S is very large of P of S at, then one of them will decide the sensitivity in that position. And the letter height is 1 plus the max of 0 or the sensitivity that was computed. But I think really you can think of other formulas to compute the sensitivity, but this is just one suggestion that was very effective. So
can you maybe repeat what is PFS? Just the
So P is the model, is the predictive model. Let's say you train the deep neural network, and now you can predict a score for any sequence. Then P would be the network.
Got it. So basically for us, P is whether it's the model or it's basically any function that we plug into this definition.
Yeah, I mean, the model approximates some function. Yeah, but any function you can use.
Thank you.
So let's look at a few examples. So here I give you the legend where red means that the mutation would increase the score, blue means the mutation would decrease the score. So here, for example, for this protein OXD13, you see that there is mostly a decrease in these positions. And here there is a flanking position, this C, that mutating it to A and G would even increase the score. So this shows that there is an effect of the flanking positions on the binding site. And these stars or asterisks are taken from two databases. of a disease-associated variants. So indeed, the fact that many disease-associated variants fall on the binding site hint that it's probably a functional binding site. Moreover, in this visualization, they added the evolutionary conservation represented by the fastcons scores. So you see that the binding site fall on evolutionary conserved regions, which also hint to the fact that they are functional. Because if they would not have been functional, they would not be conserved in evolution. So this is one binding site. And of course, the mutation map needs to be applied by different model for each protein. Here is a different protein, the NFYA. Of course, this part of the sequence, you needed to take the model of NFYA and apply the mutation map visualization here. And this is some other protein. And here are one more example for this mutation map visualization. So in my opinion, it's very effective. You can apply it to any model, irrespective, whether it's a CNN, RNN, whether it's not even a deep neural network, a classic predictive model. And it's very easy to interpret and look at and ask what would be the effect of the mutation, and can give you the regions that are more important in the sequence than others. But I think I still see one disadvantage. Maybe someone want to say what would be a disadvantage?
Well, it only charts the importance of one nucleotide at a time. Like you can't see like bigger, like how like a string of nucleotides is important.
Excellent. So indeed, that's one issue, that the mutations are done once a nucleotide at a time. And if there are interactions between nucleotides, you won't see the effect of interactions. And there are studies that try to solve this problem. But indeed, this here we won't see the interaction effect.
No, can I ask something about this one?
Yes.
Yes, because the main, as I understand, the main issue in the interpretability in the deep neural network is there are many layers we need to interpret. But this, each layer, every layer has some of meaning, as I understand, but it is difficult to choose how this interpret these many layers. In the convolution neural network, as in terms of we have many kernels, but we don't know how to how to explain these kinds of kernels or which kernels we need to choose to interpret. But how this approach can solve these kinds of issues? That's my questions.
Okay, so excellent question. I'm going, and that will lead me to the next technique I'm going to show. So okay. Thanks for the question. But what I tried to convey so far, that and your question hinted on that. For example, if you try to visualize the convolutional layer in the kernels, that's already quite difficult, and the visualization, it's not going to be that good. And the mutation map solved the problem because it is based on the prediction. So it takes all of the network into account. It doesn't separate it to layers. So you can take any model, any black box model, and all you need for this visualization is just to have the prediction.
Okay, so it's bypass some kinds of.
Yeah, it solves the problem. It treats the model as a black box. And one more advantage that I think of, that it works on a single input at a time. I think usually when we have a model, we would like to ask what are the features the model learned, what are the global features. But here, what we visualize and see is relating to a specific input sequence. And it's a different question. How do we take the mutation maps of different sequences and combine them together to learn some global pattern that the model has learned? So the mutation map doesn't give us that. It's not global interpretation, but it's a local interpretation. And now I move to what I think it's the winner of all of the techniques I'm going to show. It's still applied to a single sequence. It doesn't solve that problem, but it's really such an effective way in my experience in deep neural networks and in genomics and in general in many applications on text, on images, and that's called integrated gradients. So I'll teach you the math behind it, and then I won't go into the theoretical proofs, and then I will go into examples. So integrated gradients, it's I think the most popular interpretability technique these days in deep neural networks. It could be applied to any differentiable model, and also specifically to deep neural networks, which we know are differentiable by the way they are trained. It can be applied to images, text, structured data, genomics. It's very easy to implement, and Ophir, my student, will give you the hands-on experience in the second half. We'll show you the implementation that you can just take a function in Python. It has theoretical justification, which I will skip today, and it's very efficient computationally relative to alternative approaches. And here is an example, not taken from genomics, but from binary classification of images that contain a firewater boat and images that don't contain a firewater boat. And we would like to ask, what are the pixels in the images based on the model decided that this image contains a firewater boat? And I think what it shows here, that it mostly looks at the water jets, right? Or the water that comes out of the boat. Would you say that the model was successful in classifying water boats or not? That's a different question. Maybe because I think it missed the boat, but it did capture the fact that firewater boats eject some water out. But that was the case for this classification problem. And that's what we want. We want to see the pixels that were important for the decision the model made. So in general, what the attribution problem is and the way it is defined, we want to attribute a model's prediction on input features. So we want to get a score for each input feature, an attribution score of how much the model cared about that input feature in making its decision. So we can attribute a learning model's prediction to its features. If it's an object recognition network, like the example I just showed, we ask what are the pixels that were important. If it's a text sentiment, maybe we want to ask the model what were the words that were more important than others in deciding. So let's say we want to classify tweets to positive sentiment and negative sentiment, or an email to spam or not spam. What are the key words in that email based on which the model made its decision? In general way, why this prediction? And the integrated gradient works great. And then, given these examples, I can ask you, what are the input features we would like to attribute to in a DNA sequence?
Nucleotides?
Yeah, exactly, right? That's the most straightforward answer. If we have a sequence and we have the nucleotides in the sequence, we want to ask the model which of the nucleotides in the sequence were the important ones based on which it made a decision. And just to give credit, I've adapted the slides from this online source. Okay, so the attribution model. Let's start with, and this is general to any model. So first, I want to ask you, if I want to attribute the importance of each input feature, in some models, it's very easy to assign the importance immediately without any computational technique. Can you guess which of the models are very easy?
fully connected.
Excuse me?
Maybe when you use fully connected, and then you can just look at the weights, and that's an importance.
Right. So in a fully connected layer, right, each neuron performs a linear combination. So the linear combination assigns a weight or multiplies each input feature by some weight. So in fact, linear regression, logistic regression, which are just a neuron with an activation function, and in general, what I think the answer would be, is a linear model, right? Because in a linear model, we have a coefficient that multiplies each of the input features. So what would be the importance of an input feature? The value of the coefficient. We can also get a positive attribution or a negative attribution. So linear models are easy to solve. But then I can ask, when is it more difficult?
Non-linear.
Right. So all the rest, right? Yeah. So non-linear models, and we'll see how we deal with that. So the first solution to the problem, and that's not integrated gradient, that's before integrated gradient, is feature times gradient. So we attribute the feature importance by the value of the feature times the partial derivative of the output to with respect to that input feature. So that's a very popular method that was performed before integrated gradients. And it captures the sensitivity of the output with regard to that feature, right? Because we take the output and we derive it by the input feature. What would be the gradient in linear models?
B.
Right, exactly the coefficient, right? If I go back, the partial derivative of y based on x1 will be exactly the coefficient. So that's intuitive. And for nonlinear models, that's the first order Taylor approximation. And there were methods that popularized this approach, saliency maps, and some other methods. I've never used them myself because I already used integrated gradients, which I think is the next generation. But in papers, I think you will see many refer to this approach of saliency maps. And here, as an example of this approach, applied to the fireboat classification problem. So here we have a picture with a boat, and we ask what are the pixels that the model cared about? And are we happy with the solution here, the attribution of these pixels? I don't think it looks that good. You don't see the object of the boat. You don't see so much the water sprays, these water jets.
Looks like we're
yeah.
missing the boat in this picture. You see this black
Right,
line again.
right. Exactly. So it doesn't work that well, at least in this example. So let's see how we solve it. So gradients in the vicinity of the loop, in this case, looks like this. So the solution is the following. So what we see and was observed by people who developed integrated gradients, that the gradients in the vicinity of the input are not that informative. It's an uninteresting gradient. But if we take a picture, the same image, and bring it closer to some baseline, and I will define the baseline soon, the gradients look much better. Here, for example, you already see some of the boat, and here it becomes much clearer. So if we take the picture and make it not as bright, and we apply the gradients approach, then the gradient really shows the boat and the water coming out of the boat. So what are we going to do with this approach? We are going to take the input, and this is the interesting gradient. We are going to take the input and start scaling it from a baseline. And a baseline for an image could be, for example, what's the baseline in this case?
Just zeros, so only black.
Exactly. and all zero metrics, all black. And then we start making it brighter. Okay, it's easy just to multiply the input by some factor. And for each one of these scale inputs, we compute the gradients. And then maybe the integrated gradients gives you the answer. But what do we do with these gradients of scaled inputs?
We stop at some cumulative brightness or something.
No, we can go all the way from the baseline to the final input.
Mm.
But then my question, once we computed all these gradients, what do we do with them to generate a single attribution?
We can put them together in a linear combination, perhaps.
Yeah, even a very simple linear combination. We just sum them. Yeah, right. And the name integrated gradients hints that it's a sum, right? An integral is the sum. So the main point that was observed that people developed integrated gradients, there are regions of the scaled inputs where the gradient is interesting, and there are regions where it's uninteresting. And when they do this scaling over all, from the baseline to the inputs over different scale inputs and compute the gradients and take the integral of them or the sum, they get a good result. And let's see an example. So first of all, let's go through the formula. Here is the integral.
Can I just ask
from
a
zero.
question?
Yeah, please, yeah.
And when we're looking at pictures, it's very easy to say this is an interesting gradient or this is uninteresting because we can see. But once we go into DNA, how do we know that it follows the same path, basically? That we know that there is a certain region that is interesting, uninteresting, like that. This feels very intuitive for me, but not proven or rigorous.
Right. So first of all, the theoretical justification is given in the paper, and I skip in the presentation today. And I don't think the interesting and uninteresting is intuitive or heuristic. It's hand waving. It's not a proof. And I don't think you can prove that. I mean, the intuition is that there are some regions that are interesting, some are uninteresting. And by taking the integral, you capture enough of the interesting so that this attribution approach captures enough of the import of the signal that tells you why the model made a prediction. It's not that there is a clear deviation between interesting and interesting.
Okay.
But the main point here is that different scaling of the inputs give you different gradients. And for the theoretical justification, I will send you to the paper because I skipped it from the presentation today. I will give the theorem soon, but I won't explain further than that or in more detail.
Excellent. So I'll ask you in the questions later in the chat to send me the paper. Thank you very much.
Yeah, sure. I will give a reference soon. And also in genomics, I will soon go into genomics and ask you what would be a scale input for a DNA sequence. And also, you also hinted on a problem and asked about the problem of how can we tell what's the true, how can we evaluate interpretability? That's another issue that I will also talk about soon. Okay, so let's go to the formula. So here you see the gradients is taken over the output, right? But for an input, a scaled input. So the scaled input is composed from alpha times the input plus 1 minus alpha of the baseline. And then the derivative is taken per input feature. The inputs are scaled from 0 to 1. So let's say an image goes all the way from a black image, all zeros, to the complete input, and multiplied by the difference between the input and the base. And when it's applied here in the case of the binary classification to images that contain a water boat, you see it works much better than the original feature gradient approach. So now I will give you a short exercise. here is the integrated gradient formula. Again, it's the same formula that we saw before. We take the scaled input, which is x prime, the baseline, times alpha of x minus x prime. We compute the partial derivative based on x with respect to xi and take the integral with respect to alpha, which is the parameter that tells us how much of the input we take and how much of the baseline. And now my question would be for you, and I will give you maybe 2 or 3 minutes, is to write the integral as a sum over m bins, right? It's the, I think, Riemann's approximation for the integral. And that will also relate to a parameter that you will see later in Ophir's hands-on example. So I will give you 3 minutes, and please go ahead and try to write this formula, but approximated as a sum. Okay, so does anyone have an answer, wants to write it? Actually, you can also annotate if you want. If you want to answer and write the equation that you got. So if anyone wants to volunteer. Okay, so let's see. I will try to annotate on. I need to find the annotation option. Okay. So I have here annotation. Let's see if it works. So we take xi minus xi prime. And now we said we replace the integral with a sum. So sum over what? How many values do we iterate in the sum?
All features.
M, right? So the iteration over the features appears here in the index I. So we iterate over m bins, and each bin is going to give us a different scaling of the input. We'll still sum the derivative. So let's take f and the derivative by xi. And now here, we need to write the scaling of the input for that bin. So that will be x prime times, and what would be the factor that we multiply? X minus x prime.
Alpha m, alpha k.
So we are going to replace alpha. We are not going to have alpha. Alpha is going to be something that depends on k and m.
K by m, I guess.
Right, exactly. So that's k over m, right? In the kth bin, we take k over m of the original input, x. And is that all, or do we need something in addition in this formula?
Delta alpha. So k over m.
Exactly. We need something that exactly. It's going to be 1 over m, because for each one of these values that we sum, we need to multiply by 1 over m as we sum m of those. So that would be the approximation as a sum. So that's kind of an exercise to get you into the formula. Let me clear it. Stop the annotation. Okay, good. So now you have the formula here that we just wrote. And what would be a baseline? So the baseline, ideally, we need to decide on it, and as the users of integrated gradients, we need to pick it, is an informationless input of the model. So for example, in images, as we saw, could be a black image, all zeros. For a text, could be an empty text or a zero embedding vector, right? If we learned about embedding in NLP and text applications. And then I can ask you, what would be a baseline for a DNA sequence?
A uniform distribution.
Right, so one option is a matrix that is uniform distribution, and that's an informationless vector.
We can take reference maybe, or like reference sequence based protein.
A reference sequence, I'm not sure it's informationless.
Well, but we want to compute diff in comparison to something that doesn't carry information to us. It might be not absolutely informationless.
Right, so maybe a combination of different such baselines. Let's say you take many sequences which are informationless and compute the integrated gradients from them. That could be a good approach that we've experienced with. So following also the uniform suggestion, we can also look at the nucleotide distribution. If it's a specific genomic region or the whole genome, again, if GC content, for example, is 60% and AT is 40%, we can build a matrix that is the baseline based on those distributions. And actually, the all zero matrix works really well as well for genomics, not just for an image. So later on in the exercise that my student Ophir will give you, you will have an opportunity to try different baselines and see the effect they have on the result of the integrated gradients approach. Another issue is the scaling of the baseline to the original input. And why is that an issue in genomics? Imagine that I take the all zero matrix or the uniform matrix, and I start to build the scaled inputs in the integrated approach. What's the issue? I mean, what could be a problem? So the baseline, when it is scaled to the original input for us, a DNA sequence, as you know, is one hot encoded. But a scaled input contains values between 0 to 1. Okay, the matrix, right? Let me annotate maybe, so it will be easier to understand the issue here. But the input, usually it's one hot encoding, right? We have 1, 0, 0, and 0. But if we start scaling it from an all zero matrix, we can have 0.1, 0, 0, 0, and so on. Now, 0.1, is that a valid input to a model that predicts on DNA sequences? No, because the model only saw one hot encoded matrices. So it's not clear if the integrated gradients will work on that. And there is a method that tries to overcome this issue of a scaled inputs not being real DNA sequences. So as we said, the importance of the baseline and the choice that we make for genomics, which is the most effective one that I saw in my experience, are the all zero or uniform matrices. And let's look at a few examples after we've gone through the math and the technique. And here an example taken from a study by Uwe Euler, where they trained neural network to classify RNA binding sites. And here, for example, they classify binding site of the PAM2 protein, and you see it really works like a magic. It really highlighted the important nucleotide. This is the known motif for this protein, and you see that the motif appears quite well. And the way they evaluated it is they counted how much of the positive sequences included this motif, and they found that it was in 203 out of the 353. Another example is some other protein, where you can see that the motif GIUC is highlighted very nicely in the sequences. And it appears in most of the positive set sequences. So that's why I say it works like a magic. In my experience, it works really well. And just to note that there are many other variants for the integrated gradients approach. And this is just a very uncomprehensive selection, deep lift from Anshul Kundaja's lab, which tried to overcome the problem that I raised by the fact that scaled inputs are not real sequences. Enhanced integrated gradients from the lab of Yosef Barash, deep SHAP, which is a variant of the SHAP approach, which I haven't covered, and also a recent work that was published by Linder et al., and it's only in bioRxiv for now. And there are many other more methods called attribution methods that they assign the importance of the model for each input feature. So this is the theorem that justifies the use of integrated gradients. I will skip it for now, but those who are interested can go and read the theoretical justification. So to summarize integrated gradients, it's a technique to attribute a deep network or any differentiable model prediction to the input feature. It's very easy to apply. It's widely applicable. It's backed up by theory, and you can read more about it. And now I will proceed to the last part of my talk, which is how do we evaluate interpretability? So I gave you an example of producing kernel visualization, producing mutation maps, applying attribution methods such as integrated gradients. But now, right, when we have a new model, we have a way to evaluate it by some test set, and we have a Pearson correlation metric. And we can say that Pearson correlation of 4.9 is greater than 0.8. We get an AU-ROC for binary classification. What is the problem with evaluating model interpretability? And that's the question to you, if someone can suggest.
Well, you don't always have like a ground truth to compare to.
Exactly. The main problem is that we don't have the ground truth. I mean, what we want to learn from the scientific purpose is the biology. if we knew what the biology underlying the measurements, we didn't need to interpret, but we don't know the ground truth, right? When we have a model that predicts binding or some other property of a DNA sequence, we have some other measurements that we can compare to, which are the ground truth. Here we don't have the ground truth. So I will give you an example of one partial solution. Some of the solutions are to use simulated data sets with embedded elements. So you can take a sequence, embed specific motifs or elements in it, and then treat those as ground truth. And I will show you an example that I've done with a study with my student Ophir, which you would also go over in the hands-on exercise. And that's to perform an experimental validation on sequences that include embedded elements. And maybe you can suggest other ideas. I'm open to suggestions. Okay, so now I'll go into our study and how we evaluate it. And that's one of the novel parts of our study, I believe. And you will also see this in Ophir's presentation and exercise. So the application that I will show you deals with the modeling and prediction of mRNA dynamic and rates. And let's say for simplicity that we are given an RNA sequence from the 3' UTR, and we need to predict how quickly the RNA is going to degrade. As we know, RNA are unstable molecules. We all know about the mRNA vaccine. We need to keep in very cold refrigerators, freezers. So an RNA is an unstable molecule as opposed to double-stranded DNA, and it has some degradation rate that depends on sequence element inside it. So the input is an RNA sequence, and the output is a number that is the degradation rate. Training data is quite nice. It's taken from the 3' UTR-seq dataset published by Michal Rabani back when she was a postdoc at the Broad Institute. It includes 90,000 sequences, each of length 110 nucleotides. And following this original dataset based on which the training was done, Michal took the strongest k-mers from a lasso model she trained and used these strong k-mers to embed elements in a set of validation sequences. So a follow-up experiment was performed on 58 sequences, synthetic sequences, with these embedded elements. So for us, this is a nice benchmark because now the ground truth are known, now the embedded elements. We need to pick them up. And here is an example of a sequence. And with the gray shadow, you see the embedded elements. So imagine you have a sequence and you can really insert embedded elements and then perform an experiment to see the degradation of that sequence. And now I show you the application of integrated gradient on this approach. So the integrated gradient gives us an attribution, the importance of each nucleotide in the sequence. And as you see, the CNN model on this training data really was able to pick up these two elements, I think, quite nicely. And we have two versions of the model. One is a single task and one is multitask, which maybe Ophir will go into details in his presentation, but it also performed well in picking up these two elements. We also have a recurrent neural network model that kind of missed the first element. You see that there is no strong letters in the first element, but it did capture the second element and also the multitask version of the problem. And the known elements are a microRNA binding site, an A-rich element, and the site. So here you get to see the microRNA binding site, and this is the A-rich. Another example includes a poly-U rich element, poly-U, so it has many U's. T is on the DNA level, but U on the RNA level. And also the CNN, you see, highlights this poly-U element much better than the RNA model. So we suggest the following way to evaluate the interpretability. It's something that we came up with, and we also, Ophir will suggest another metric. This is the following. We take a sequence, we apply the integrated gradient approach on it, and we get the attribution metrics. Then we sum up the values for each of the position. We get an attribution score for each position in the sequence. We also have a ground truth vector with the denoting +1 for stabilizing and -1 for destabilizing. Just to go back, here you see that some elements have a positive effect, meaning they are stabilizing, and some elements have a negative effect. And this is captured by the integrated gradients approach. So we can assign +1 or -1 based on what was known from prior knowledge or based on the last model in terms of the effect the element has on the degradation, and 0 for any other nucleotide position. Then to measure or to approximate or evaluate interpretability, we compute a Pearson correlation between the ground truth vector and our attribution vector. And this is the interpretability quality score. And here you see over the 58 validation sequences that the CNN models, the convolutional neural networks, are performing much better than recurrent neural networks. This is based on, there are two data sets, A- and A+, so here we'll get into the details. And we saw the same trend for the A+. We can't say if the multitask or single task is better, but we can clearly say that convolutional neural networks are much easier, at least with the current techniques we have, are much more interpretable than the recurrent neural networks in our applications. And while recurrent neural networks can be a bit more accurate in prediction, still the CNN are just slightly behind usually, but they have much more interpretability, and we show that in our case. Now, the problem that I raised before with the mutation maps, and I think I have only a couple more slides before I conclude, is the fact that the integrated gradients or any attribution method is applied to one sequence. So all of these pictures give us local interpretability. What was important for a single sequence as decided by the model. But it's not a global feature or a global pattern that the model has learned. So the question that we need to solve, for example, if we have many such pictures, many such attribution maps, what are the common patterns? And I know of one solution to this problem, which unfortunately, due to the time limit, I will not go over today, which is an algorithm from Anshul Kundaj's lab in Stanford called TF Modisco, developed by Shir Kumar et al. So you can go later and see it, and Ophir maybe will also talk about it a bit in his presentation, or at least you will have a code example on how to run. But the goal of TF Modisco is to take the attribution matrices, attribution maps, and find the common patterns, and then to give a global representation of what the model has learned. So I would like to conclude with the following slide of the open problems that I see in the field. I think it's a very active field, the interpretability of deep neural networks, and many researchers are still diving into it, and it's still a problem. How do we solve this black box nature of deep neural networks and learn some biology from it? So first of all, what is the best attribution method? I've shown you just one method, integrated gradients, but I have also listed a few other: deep lift, enhanced integrated gradients, deep SHAP, and there are many more. I think more than 20 or probably more that I don't know of. And one of the key solutions to answer this question is that we need a benchmark to evaluate the interpretability. Just producing a benchmark is already a problem by itself. As I said, we don't have a ground truth of what is the real biology. So we need a benchmark, then we can evaluate and pick up the best method. And in addition, in all of the examples that I talked about, I only talked about the importance of the sequence features, the nucleotides. How do we assign importance when we combine different features in the same input, such as RNA structure, epigenetic information, evolutionary information that can be provided as input to the model, and give us a more predictive power. So also this question comes up. And as I told you, finding common patterns in attribution maps. Currently, I know of only one solution to the problem, which is TF-Modisco, but is it optimal, or could there be other solutions that are better for this problem? I mean, maybe it's an NP-hard problem. I don't think there is still a proof of complexity, but if it is similar to the motif finding problem, then it's probably NP-hard, and then we can have many solutions, which we'll have to pick up the best one or the most useful for each application. So with this slide of open problems, I would like to conclude the presentation. Ophir will cover what I told about integrated gradients and the effect of the hyperparameters on it in his hands-on exercise. So I welcome any questions or discussion if there is time.
Yeah, I think we have time for questions. Please feel free to ask. You can unmute yourself, you can write in the chat. Yeah, thank you so much. It was extremely interesting and indeed a new step forward, this integrated gradient technique and its application to genomics. It's quite an advanced knowledge to all of us. Thank you so much. Well, probably people can ask questions during the seminar. If there is no question, we have a short break till 30 minutes and start seminar in about 7 minutes. So we follow the schedule. You are staying with us or you are leaving?
Yeah, I'll be here. Yeah, so the schedule indeed said that I should talk till 12:20.
Yeah.
So that's why I concluded 3 minutes late. Yeah.
Yeah. So so you still can continue answering questions during the seminar. Yeah. So let's have a short break for now.
Maybe I will share my presentation.
Okay, so let me stop share.
Okay, let's continue. 30 minutes sharp.
Okay.
Who will be held in the seminar? Yaron, you or somebody else? Or if
Ophir. Yeah.
Ophir.
Yeah.
Nice to meet you.
Nice to meet you.
Yeah,
Do you hear
please
me
go
well?
ahead. Please go ahead. We see your screen.
Do you hear me well?
Please go
Yeah.
ahead. We see you, we hear you, we see your screen.
Okay. Wait a moment, I will adjust my. Do you hear me right? Okay, I think I will start now. So as said, my name is Ophir Yaish, and I'm a student of Yaron Orenstein. So before we will go to have some experience with the model interpretability tools, I will present our work called computational modeling of mRNA degradation dynamics using deep neural networks. All the parts that we will see in the end-on section are based on this work, the dataset, the models, and so on. So let's start with the motivation of this project. It will be a little bit not a lot of informative, but probably most of you know all of these things. So as you know, all the genetic code that characterize the organism is kept in the DNA, and in transcription stage, part of the DNA are coded into RNA. And in fact, those RNAs are expression of specific part in the DNA. And in the process translation, mRNAs are translated into proteins, and of course, those proteins are a key player of the organism cell as they carry out of the operation that are vital to the existence of the cell. So, as Yaron said, the RNA is extremely unstable molecule, and its stability is regulated according to its sequence, structure, and the other parameters. So in our study, we are dealing with the mRNA degradation, and understanding the process of this degradation will help us to understand the stability mechanism of the cell, essentially. So we have two main goals in this study. The first one is to build a computation model based on deep learning, which will model the mRNA degradation dynamics. And the second one is to obtain biological insights on the degradation process using the interpretability tools that some of them Yaron explained before. So now I'll go briefly over the dataset. The dataset is taken from a study called this long name. I will not repeat it again, and I think Yaron said it, by Michal Rabani. So in this study, they performed an experiment on zebrafish embryo cells, and they injected the synthetic mRNAs during the embryo development. And during the development, they measured the mRNA abundance in different time points. Essentially, when we are saying that the abundance is decreasing, the mRNA 11 is decreasing, it means that there is degradation of the mRNA. So the dataset is comprised of the mRNA sequences and their corresponding mRNA levels. Here they are. As we can see in the first table, each sequence is 110 long. In their study, they created two datasets, one called A- and the other one called A+. Each one of size 90,000. So the difference between them is that in the A+, the mRNA should be more stable since they add poly A to the flanks of the sequence. Not the flanks to the 110 long sequence. So in terms of the 112 long sequence, both A- and plus have the same sequence. And now about the mRNA levels, as we can see in the second table here, if for each sequence, they sample the mRNA levels at 9 different time points, starting from the first tower to the 10th tower with the exception of the 9 hour. So here we have a different measurement for the A- and the A+, of course. Since the A+, as I said, should be more stable. So the main assumption of the previous study is
assuming by
that,
normalization you mean
I don't hear you. Can you repeat again?
can you just say what do you mean by normalization? What is it normalized against?
I think that you got to read counts after doing high throughput experiment like that, and then I don't know in this experiment how it was done exactly, but sometimes it's normalized by the maximum read counts or something like that, and they also logged it. They took the log of the mRNA level of the abundance. I think
Excellent, thank you.
So, as I said, the main assumption of the previous study is that the dynamics of the mRNA degradation is linear or piece, or more precisely exponential, as you can see, and the log of it, as I said, is linear or piecewise linear if we're assuming that the rate change as function of time. So in the study from which we took the data set, they suggested 4 linear functions, as you can see in this figure. And for each sample, they choose the one which more likely to describe it, choosing the less complicated one at first, and then if it's not fit, they're going to more advanced one. So now one of
Phil, we can't hear you.
Because we're not fitting a threshold on experiment parameter called number of reads, as we said. I think I lost. Do you hear me now?
Okay, yeah, you are back to the good mic. Yeah.
I think something happened. I heard the microphone say something to me. So I will repeat maybe when I stop.
Yeah, just start from this slide.
Okay. So what I tried to say is that one of the issues that we had with previous studies is that they used only a third of data, and because the reason for that was because some of them were not fitting well their linear model, and because some of them were not fitting a experiment parameter called number of reads. They got lower value of lower number of reads than a threshold. Certain threshold that they choose. So what we try to understand is how well their fit, the 3' mutar fit, is good for all the data set. Since we had their fit only for a third of the data, we had to split our examination into 2 parts. In the first part, we had to convince ourselves that a simple linear fit is as good as the 3' mutar fit. So as you can see in this figure, the average R squared of both fits, the 3' mutar model fit and the linear model, are almost the same, and therefore we conclude that the linear regression model is as good as the fit with the model with different starting points or something like that they had. So in the second part, we checked how the simple linear model fits all the data set. And as we can see in here, the average R squared is now much lower than it was on the linear set. And the significant subset of the reporter degradation dynamics do not fit the linear model. So next we continued our data exploration and tested the correlation between the mRNA levels at different time points for the A- and A+, and we saw that in both A- and A+, the mRNA levels at different time points are highly correlated, starting here of around 0.8 or something like that. So combining this knowledge with the fact that the linear fit is not good enough, we developed a new approach to predict the mRNA degradation dynamics given the input of the mRNA sequence and its initial mRNA level, the 1 hour mRNA level. Given those inputs, we predict the mRNA levels, the rest of the mRNA levels, 2 to 8 and the 10th hour. So we just need to process the sequences into features and choose the right computational model, the right machine learning model. So before I will describe our deep learning models, I will talk about the machine learning models which will take part in the performance comparison. So we created 2 models that were used in previous study. The type of the machine learning is the same as it was in the previous work, but the difference is the inputs and the output, of course. They said in previous study, the outputs are the degradation rate, and here are the, in our study, the prediction, the outputs are the mRNA levels different time points. So as I said, we have to process the sequence into features. So like in previous studies, the sequence feature has the k-mer composition, meaning that we are counting for the number of times that any possible k long sequence occurred in its sample. For example, we can see in this figure that all the 4 formers of the sequence above, and for each former we can count how many times it appears in the sequence. So the first model is called lasso regression. It's like a simple regression, but with L1 regularization. This L1 regularization help this model to do models feature selection, and we will see in our examples how it's this is very benefit to the model interpretability of this model. The second model is called random forest. It operates by constructing multiple of decision trees and outputting the mean of prediction of the individual trees. And the settings of those models were the same as reported in previous studies. So let's continue about now. Let's talk about the deep learning models. So one of the most important advantages in deep learning, as you probably heard in the last lectures, in the previous lectures, is that we can learn from the data directly without a lot of feature engineering. For example, in the machine learning models that we saw earlier, we had to create the k-mer counting, which shall not describe the sequence properly. It's not one-to-one function from the sequence to the k-mer counting. In deep learning, we can transform each sequence into a simple one-hot encoding metric, as you can see in this figure. And this is those will be the sequence feature of the models, the input. So before we will go to talk about the architecture of the models, I will say some word about their foundation stones. You probably know that. So the first one is the one-dimensional convolutional network, 1D CNN. It's similar to the 2D convolution, but the difference here is that the kernel is moving only in one direction. And therefore it's used mostly in a time data series or data like we have here, series data. So what's special in this layer is that is good in extracting spatial features of the data. And this is why we think that it had better performance in the interpretation than the RNN model, for example. The second foundation stone is, it's called the recurrent neural network, RNN, which is highly used in time series data as well, or language for data, which is similar to what we have here. And the reason for that is that as you can see in this figure, you got this block that that work iteratively. I mean, and when we are unroll this loop, we